Leticia Moreno-García. She's a PhD student in Rosario Osta's group and at Langevio and University of Zaragoza, and is a group of CIBERNED. Her poster is number 10, and she's going to talk about circular RNAs as blood-based biomarkers of ALS. Thank you.
Good morning. Thanks for the kind introduction. As we all know, ALS is the third most common neurodegenerative disease and the most frequent of motor neuron diseases. A significant increase of ALS cases has been predicted, and there may be 69% more cases in 2040 than 2015 due to population aging. This is an alarming fact that also does not take into account the diagnostic delay, which is usually more than 1 year. Therefore, there is an urgent need to identify sensitive biomarkers of ALS. To date, the most reliable biomarkers proposed for ALS are proteins. However, RNA biomarkers are another possibly cheaper alternative. In fact, aberrant RNA metabolism is a key factor in ALS pathogenesis, so RNA biomarkers are a good option for use to diagnostic and prognostic ALS. Between the different types of RNAs, there is one group that is especially stable due to their structure. This group are circular RNAs. Although RNAs have hardly been studied in ALS, it has been seen that they are powerful players in other neurodegenerative diseases. This could be due to their great influence on the CNS, since neuronal tissues, like the brain, present a higher concentration of cRNAs and their levels increase with the aging. cRNA present important functions, including the regulation of gene expression, either through their interaction with RNA polymerase II, their binding to microRNAs or RBPs, and by the regulation of parental gene splicing. In addition, cRNA present advantages over other types of RNA, such as a longer half-life. They are present in exosomes and biofluids, and they are highly conserved across species. All this makes them ideal biomarkers candidates. So, in this context, our objective was to assess the diagnostic and prognostic value of cRNAs as blood-based biomarkers of ALS. To this end, a total of 15 healthy controls and 19 ALS patients participated. On one hand, the functional status of ALS patients was estimated using ALS-FRS-R scale, in which the lower the score, the worse the functional status. On the other hand, peripheral blood samples were collected in packaging tubes from both groups of patients. Then, circular RNAs were isolated. At this point, we decided to study hRNAs, and how did we make the selection? Well, first, we selected RNAs that we had previously studied in our mouse model of ALS, the SOD1 transgenic mice. In previous years, we performed RNA sequencing studies in spinal cord samples of wild-type and transgenic mice at asymptomatic stage. Next, we analyzed the expression levels of the most important cRNAs during the disease progression in spinal cord as well as in skeletal muscle. And finally, we obtained the homologous cRNAs in human by sequence alignment. And second, we selected cRNAs related to neurodegenerative processes through a bibliographic search. Once cRNAs were selected, we performed retrotranscription and real-time PCR. To compare the expression levels between the two groups of patients, we used students' t-tests, and to determine the correlation between the levels of cRNAs and the ALS-FRS-R score at sampling, we used Pearson correlation. So the results of the present study can be divided in two parts. The first part allows to analyze the diagnostic value of these RNAs, and the second, the prognostic nature. Regarding the first part of the results, 3 of the 8 RNAs studied were differentially expressed in ALS patients with respect to healthy controls. Specifically, 2 RNAs were statistically upregulated and 1 downregulated. Next, we further analyzed the diagnostic value of these RNAs through ROC curve analysis. The 3 circular RNAs presented a considerable predictive power since their areas under the curve were over 0.75. By combining these 3 circular RNAs, their predictive power increases with an AUC value over 0.9. So the panel of these 3 circular RNAs could be a potential diagnostic tool. Regarding the second part of the results, a total of 4 significant correlations were observed between delta CT and the ALS scale. These results suggest that the lower the abundance of these RNAs in blood, the worse the functional status of patients. CRNA 3 presented the strongest correlation with the functional status of patients. It presented a Pearson coefficient of 0.72. Interestingly, this RNA comes from a gene that codes for a protein which is involved in ALS pathogenesis through nuclear transport. In view of these results, we can conclude that 6 of the 8 RNAs studied showed a potential diagnostic and/or prognostic nature in the peripheral blood of ALS patients. Of these, RNA4 could be a biomarker for the diagnosis of ALS as well as the prognosis of functional status of patients. These results are highly relevant since this is the first time that is analyzed the RNA expression in whole blood of ALS patients, including all blood cell types, which may be of help in clinical trials and practice. Nevertheless, more exhaustive studies are needed with increased sample size in comparison with other neurodegenerative diseases. Considering all the limitation of this study, we are already working in the following 3 steps. First, increase the number of cRNAs. Second, increase the number of samples with patients of other neurodegenerative diseases, as well as serial samples of ALS patients in order to do a longitudinal study. And third and last step, study these cRNAs in plasma samples to further facilitate the process. Finally, I would like to thank all the members of la Bio group in Zaragoza, Cristian Luneta, Silvana Penco, and Claudia Tarlarini from the Niguarda Hospital in Milan, Alberto García Redondo from 12 de Octubre Hospital in Madrid, as well as all our funders. And thank you so much for your kind attention.
So thank you very much, Leticia, for adjusting so well to the time. We have 2 minutes to go for questions. No questions? I have one myself. So how big is the sample you have used for this first study? What is the number of patients that the samples you analyze? How many? 19 patients. So you want to increase the number so that you can consolidate your results. Okay, thank you very much, Leticia. We go on. So now I have to present the very experienced but forever young researcher, José Luis Lanciego, that is at the CIMA, University of Navarra, and at CIBERNED as well, a principal investigator, poster number 21. And he's going to talk about blaming neuromelanin for Parkinson's disease, time-dependent tyrosinase overexpression drives endogenous synucleinopathy in non-human primates. Thank you very much, José.
Good morning, everyone. Thanks a lot, Teresa, for such a lovely presentation. As you can easily realize, I'm happy to be here in the session for young trainees. I feel myself even younger than ever. So, well, these are my disclosures, but what I'm going to talk today is unrelated, so it doesn't matter. So, well, I think that all of us in the room, we will agree that we need better animal models when coming to study neurodegenerative disorders, Parkinson's disease in my case. And when coming to model a neurodegenerative disorder, you need to keep in mind what exactly do you want to model. And we need better models mimicking the non-neuropathology of disease to the best possible extent in order to increase the chances of new drugs when going through the clinical trials. So what we know for sure in Parkinson's disease is a disorder where you are about to lose dopaminergic neurons, and you are about to lose pigmentation in the substantia nigra because these neurons have neuromelanin. On top of this, for the neurons that remain alive, there's a pathological characteristic feature that these neurons have Lewy bodies. That is the pathological criteria of diagnosis for Parkinson's disease. And both things together create like a perfect storm. And once the neurons die, there is a pro-inflammatory scenario made up of microglial cells and peripheral macrophages. So here you have some examples of Lewy bodies in the nigra. These are positive for alpha-synuclein. Those are positive for p62. That is a very good marker for Lewy bodies. So we were thinking about neuromelanin for a while. Just in between, this lovely paper from Miquel Vila arrived. Miquel, you know him for sure. He's a super smart guy. So Miquel has managed to create an AAV encoding for human tyrosinase gene. It's the enzymatic precursor of neuromelanin. So when delivered into the substantia nigra of rats, the substantia nigra became pigmented to get a very nice series of neuropathological findings. So overall, the idea was to upgrade this rat model up to non-human primates. That for us is the gold standard on PD. So the strategy we have followed is just we deliver the AAV coding for human tyrosinase directly to the substantia nigra by means of stereotactic surgery. Once these neurons became transduced, they start to express neuromelanin in a time-dependent manner. At some point, they have Lewy bodies, and once these neurons die and neuromelanin goes extracellular, there is, a pro-inflammatory reaction. So this is, in case of success, what we want to do. So this is just a summary of the results. You have more data in the poster. As Professor Pove showed yesterday, we can keep track of ongoing changes by taking advantage of neuroimage endpoints. So here you have a T1 sequence of MRI. This is a structural MRI, where you can see in the ventral mesencephalon, this is the point of delivery of the tyrosinase gene into the left substantia nigra, and on the other side, more or less about here, you have the injection of the control vector that is an empty AAV on the right substantia nigra. If we switch to neuromelanin sequence, you can see that in the substantia nigra, when we have injected the tyrosinase coding vector, there is a reinforcement of the signal. Here, this is the typical signature for neuromelanin in the monkey brain, and there is nothing else on the contralateral side. Another neuroimage readout are micro-PET scans. It's a more functional readout. We use the dihydrotetrabenazine as a radio tracer. So you can see that, on the left side of the brain, compared to the control side, there is a reduction on the uptake of the radio tracer, both in the caudate and in the putamen nucleus. Well, once we perform the necropsy of these animals, these are images taken with my iPhone on the microtome. We realize that we have pigmentation in the nigra that is visible macroscopically to the naked eye. So this is the expression of neuromelanin in the injected substantia nigra that is more or less at the same level of what will you expect to see in an elderly human individual. Indeed, this is for an animal that has been transduced with a follow-up of 4 months. If you wait a little bit longer, this is a female animal with a follow-up of 8 months. You can see there is a depigmentation of the nigra that accounts for a progressive dopaminergic lesion. This is the typical histological image. It's contrasting with ultra-red. There are plenty of neurons that are all dopaminergic that are expressing neuromelanin. And the contralateral substantia nigra is completely empty. And later on, we went to the confocal microscope. So for the images on this panel, what you have in green are dopaminergic neurons, I hope you can see neuromelanin all around. And there, with immunohistochemistry for p62, we can disclose the presence of Lewy bodies inside these neurons. So in order to further be sure that these are true Lewy bodies, we have performed some additional stains with alpha-synuclein and ubiquitin. So for instance, you can see here a neuromelanized neuron that is Th positive with a Lewy body positive for p62, as well as for alpha-synuclein. So this is work done at the University of Santiago de Compostela in order to have some ideas of another partnering crime, in that case, the microglia reaction. So what you have in green is a stain for IBA1. This is a stain for macrophages, a positive for CD68. And here, high magnification, you can see neuromelanin. This is extracellular neuromelanin that has been digested by microglial cells and macrophages in an attempt to get rid of extracellular neuromelanin. And last but not least, we have, for this cohort of animals, we have 2 animals with a follow-up of 8 months after the delivery of the tyrosine gene. So we realized that it might be a good idea to take a look at the frontal cortex, and these images were taken at the level of the superior frontal gyrus. So what you have in green are layer 5 pyramidal neurons in the superior frontal gyrus. What you have in blue are Th positive fibers, and what you have in red are p62 positive Lewy bodies. So you can see that neurons in the pyramidal cortex receiving dopaminergic innervation start to exhibit Lewy bodies. As you can even see, for instance, in this image, this is a fiber that is positive for Th, making a presumptive contact over there, and the Lewy body is just opposite in the postsynaptic element. So this accounts for a prion-like express of endogenous alpha-synuclein, because we are not dealing with alpha-synuclein, we just enhance neuromelanin production. So the synucleinopathy we have induced here is 100% endogenous. So just to summarize, is this on time? Well, it's still preliminary, but we have prepared a non-human primate model that is quite very close to the true neuropathology you can see in human cases of Parkinson's disease. There is an important concept here, is that, as for instance, Professor Tolosa mentioned yesterday, there are plenty of companies targeting alpha-synuclein or considering alpha-synuclein as a primary target. However, by going through this procedure, we are in a position of demonstrating that alpha-synucleinopathy is triggered by neuromelanin accumulation. So it's a secondary phenomenon. First, you need to aggregate neuromelanin, and synucleinopathy comes later on. So for these trials that are ongoing targeting alpha-synuclein, I think, in my opinion, they are going to fail, because if you are about to target synuclein, it's already too late. You need to go a little bit earlier in the process. If you can find any therapeutic target, turning down neuromelanin production, for sure, you will get rid of the subsequent synucleinopathy. This is the first evidence of Lewy bodies in non-human primates, and this is just a spread of endogenous alpha-synuclein in a circuit-dependent manner. So I think these data are robust enough to provide experimental support for the drug hypothesis. And just to conclude, this is my team, made of very young people, our funding agencies, and I want to stress the fact that these results are coming out of an intramural collaborative project of CIBERNED, led by Miquel Vila, and we spent a couple of years in dealing with this. It was a couple of lovely years working with such a nice group of very close friends. So that's everything. So if you have any questions, please go ahead. Otherwise, you can come to the poster, and this is 21. Thank you very much.
Thank you very much, Lucky. So Carlos has a question. Carlos.
Well, so far we don't have any motor phenotype, because we are making just unilateral lesions. Monkeys are very efficient in compensating unilateral results. So we have now 2 more cohorts of animals that underwent surgery just a month ago. And in that case, we adopted a different strategy. We switched from an AV1 to an AV9 that is much better suited for CNS delivery. then we can perform bilateral injections with these AV9 coding stereoisomers. So we expect that the level of lesions that we are about to induce over time are going to be high enough to have a motor phenotype.
Okay, thank you very much, Lancy. Thanks for your talk. We go on. Our next speaker is Marta Garo-Pascual. She's a PhD student in Brian Strange's lab, and the group is at the Fundación CIEN and Center of Biomedical Technology at the Universidad Politécnica de Madrid. The poster is number 35, and she's talking about a hippocampal-anterior thalamic circuit associated with learning rate in aging. Thank you.
Thank you. Thank you for the presentation. So yeah, I'm talking about aging, which is not a neurodegenerative disease, but a brain also changes during aging. Here in this slide, we can see the volumetric changes in different structures within the brain for a control group of healthy agers, but also for Alzheimer's disease or mild cognitive impairment. The changes are different, but they also occur in normal aging. During aging also happen cognitive changes. Here we focus on memory, and in this plot we see how memory declines also with aging, but still within a normal range. But what I want you to draw your attention here is what we use as a memory proxy. Normally we use the retrieval component of memory tasks. The retrieval component is a delay recall score, but on this study that I'm presenting today, we focus on the learning rate. Learning rate within the 3 consecutive trials that we do in this memory task. So I'm going to go into many details about the memory task that we use. The memory task we use is the free and cued selective reminding test, also called as FCSRT. This test starts with the presentation of 16 words. Those words belong to different semantic categories, but they are not the most obvious element of each category. And after 20 seconds interference, the first immediate recall trial starts. Subjects are asked to recall freely as many words as they can remember. But for those words that they cannot remember, the instructors gives them cues that are the semantic category to trigger more recalls. This same protocol is repeated on a second trial with the same dynamics, and again on a third trial with the same 20 seconds interference. But after this third trial, there is a longer delay, a 30 minutes delay, and then a delay recall is performed. And this is what we usually use as memory proxy. We call it here retrieval. However, in this study, we focus on the free recall trials, and out of the 3, we calculate the learning rate. So in this study, we are interested in isolating how fast subjects are able to learn within the task, regardless of how many words they are able to recall by the end of the task. The objective is to study this learning rate in a healthy older cohort, and also find the neural correlates of this learning rate by studying first gray matter volume, but also white matter microstructure. Our sample is the Vallecas project sample. So this is a longitudinal study that takes place at the Alzheimer's Center, Fundación Reina Sofía, Fundación CIEN. Despite it is a longitudinal study, for this study we only focus on visit 1, and we exclude those subjects who have signs of cognitive impairment, and we also make sure they have an MRI scan available to study the brain. So we end up with 982 subjects. And these are the demographics of our sample. They are on average 75 years old, 65% women. But I want you to maybe look more into the increase of the number of words that subjects on average do for these consecutive 3 trials that we use actually to calculate the learning rate. So on average, subjects are able to learn 1.4 more words each trial. The first graph is just a representation of what I just told you, the increase over trials of the number of words that subjects can recall. The second graph shows that there is a negative correlation between age and the learning rate. I want to again emphasize that here we're trying to isolate learning rate, so we are always controlling for this retrieval. So we are interested in how fast they learn, regardless of the number of words they are able to retrieve by the end of the test. Also, women are faster learners, and also higher education is associated with faster learning. So now the neuroimaging correlates of this learning rate. What you are looking at is a template of a brain, and we project our significant results on top of that template. So we see there is a positive correlation between learning rate and gray matter volume in the hippocampus, very clear here. The effects are bilateral, but more strong on the left side. We also have effects on the thalamus, and within the thalamus, the anterior thalamic nuclei is the nuclei with a stronger effects. So hippocampus has a known role in memory function, but also does the anterior thalamic nuclei, and actually these two structures are well connected with each other. We also study white matter integrity, and we studying the diffusion properties of white matter of water molecules. Water molecules actually diffuse quite well within white matter, because white matter is just a system of tubes. When we study diffusivity, we focus especially on these two parameters. Fractional anisotropy is high when there is a long preferred axis of movement for water molecules. In this cross-sectional cut of white matter tract, the preferred axis of movement for water will be the perpendicular direction to the screen. And we also have mean diffusivity. Mean diffusivity will be high if water molecules can move on the same plane of the screen. Here, mean diffusivity is small because there is a very compact white matter microstructure. But in aging, this microstructure loses integrity, so mean diffusivity increases, and no longer this perpendicular direction is the most preferred, so fractional anisotropy decreases. So with this bit of context, now I can show you where there is a correlation between learning rate and white matter integrity. So in the left panels, we see the correlation, the positive correlation with fractional anisotropy and the negative with mean diffusivity. You see the correlation is in many white matter tracts within the brain, but here on the right panels, we highlight the fornix that you can see as this white skeleton, and the color regions are where there is a correlation with learning rate. So actually there is a circuit here. The fornix is the white matter tract that connects the hippocampus via the mammillary bodies to the anterior thalamic nuclei, which are the regions that we've shown in the gray matter analysis. So to wrap up, I wanted to again highlight that retrieval, which is the component that we normally use for studying memory in aging, is of course sensitive to age-related changes of memory, but also is the learning rate. So maybe this is an alternative component that could help us to better differentiate what is a normal decline in aging and what is a non-normal, so associated decline, neurodegenerative associated decline in memory. Learning rate is also associated with some brain structures that are playing an important role in memory, as we've seen the anterior thalamic nuclei, fornix, and hippocampus. So since the cognitive impairment detection is what happens, it happens very late after the cascade of events in an Alzheimer's disease process, for example, maybe it's worth exploring different proxies for memory that could maybe help us to early detect what is neurodegeneration in memory. So that's all from my side. I want to thank my co-authors, especially Daria, who I started co-authorship with, but also Pablo, Belen, Noam, Lyndon, Brian, and also Miguel. Also the funding sources, collaborators are volunteers of the Vallecas project. And yeah, we're happy to share that we have this project or this study already written, so you can check more details on the preprint if you scan the QR code. So thank you.
Thank you very much, Marta. Is there any question, or we go further? If you want, you can go to Marta's poster and ask her and discuss with her. Thank you very much, Marta. So our next speaker is Iván Burgueño, a PhD student in the group of Alberto Rábano at the Fundación CIEN. His poster is number 39, and he's going to talk about neuropathological variability in Alzheimer's disease in search of distinct phenotypes of tau pathology. So thank you very much.
Thank you.
You can start.
Heterogeneity in Alzheimer's disease may be relevant from a pathogenic, therapeutic, and diagnostic standpoint, and and the variability of the disease derives mainly from the presence of co-pathologies and the intrinsic variability. Atypical forms of Alzheimer's disease, particularly prevalent in early onset forms, are well known in their clinical and their neuroimaging aspects. On a large study, in a quantitative study on a large series of patients from Murray et al., we identified these atypical forms in a principal axis of variability of tau-positive tau pathology. And they see the two forms, the hippocampal sparing and limbic predominant forms. Along this variability axis, we can see the predominance of tau, of neurofibrillary tangles in neocortical or hippocampal regions, and was associated with specific demographic and clinic patterns. Although the original algorithm did not yield equivalent results in a similar study by another group, but a recent study based on p-tau neuroimaging identified these atypical forms with another subtypes. The objective is we present preliminary results of this of the application of a modified version of the original quantitative method by Murray et al. In our cohort, in the Vallecas study. We start with 167 brains with 82% of females, 8 of them 87, cerebral time 12, and the neuropathology of the left hemibrain has studied this brain, Alzheimer's pathology, Lewy type pathology, vascular pathology, and the other pathology. Then we exclude the brains with BRCA5 and the brains with advanced hippocampal sclerosis. Now we select the brain regions and areas in temporal, parietal, temporal, and hippocampus with 80-100, and then we do the quantification of neurofibrillary tangles. Here is the lateral temporal cortex and the steps of the quantification of cell profiler. And last, we do medial temporal lobe atrophy index. In the brain slide, the height and area of lateral ventricle in green and the hippocampus was measured. We do the analysis of quartiles of cortex and hippocampus ratios of neurofibrillary tangles in the selected 58 brains. We apply the modification of the original algorithm of Murray to be less strict, and we do 3 phases. Phase 1 is quartiles, all 3 hippocampus cortex ratios. Phase 2, at least 2 hippocampus cortex ratios, and phase 3, phase 2 mass median criterion. The phase 3 is the most similar phase of similar to hippocampal sparing and limbic predominant, and we can see here the group 1 of phase 3 has more neurofibrillary tangles in cortex, and the group 3 has more neurofibrillary tangles in hippocampus. The 3 groups we obtain, the group 1 is similar to hippocampal hippocampal sparing and has higher neurofibrillary tangles, cerebral time, medial temporal lobe atrophy, BRAC-tau, BRAC alpha-synuclein, and ARTAG in the cortex. The group 2 is typical Alzheimer's disease, and group 3 is similar to limbic predominant and has higher neurofibrillary tangles in hippocampus and number of females, APOE4, CA1, neurofibrillary tangles in hippocampus, age of onset and death, and ARTAG in medial temporal lobe. And the conclusion of the study, the modified algorithm of the quantification of neurofibrillary tangles identified 3 subgroups in patients in terms of sociodemographic and clinical profiles that are similar to previously described subtypes of Alzheimer's disease. However, in these subgroups, cerebral time and medial temporal lobe atrophy displayed opposite trends to those considered characteristic of the algorithm of the atypical subtype of Alzheimer's disease, hippocampal sparing and limbic predominant. High prevalence of medial temporal lobe copathologies may explain the higher medial temporal lobe atrophy indexes observed in our hippocampal sparing group. The exclusion of patients with full hippocampal sclerosis from the analysis might hinder the identification of patients with the expected features of limbic predominant Alzheimer, and the demographic profile of our series may hamper the identification of a patient group with the expected full features of the hippocampal sparing Alzheimer's disease. Identification of neuropathological phenotype subtypes of Alzheimer's disease is highly dependent on the demographic and clinical features of the patient series under the study. Thank you.
Thank you very much, Ivan. So we go on with Montse Soler. She's a postdoc in the group of Joan Comellà at the Vall d'Hebron Institute of Research and the Universitat Autònoma de Barcelona. and CIBERNED IP. Her poster is poster number 41 and entitled FAME-L as a modulator of tau pathology in Alzheimer's disease and other tauopathies. So thank you very much, Monse. You can start.
Thank you, Teresa. And thank you, the organizers, for allowing me to present our work in progress. And at this time is the study of FAME-Long as a modulator of tau pathology. So as you know, Alzheimer's disease is a progressive neurodegenerative disorder that accounts for cognitive impairment and brain atrophy, mainly due to cell death. Its main histopathological hallmarks are A-beta deposits and neurofibrillary tangles that are composed of aggregated tau. There are several post-traductional modifications that account for higher aggregation of tau or its loss of function, that, for example, are phosphorylation, ubiquitination, acetylation, or cleavage by caspases, and the regulation of these modifications or how they contribute to the tau aggregation are not fully understood. However, we know that neurofibrillary tangles in tau pathology is more associated than A-beta with dementia at the clinical level and with neurodegeneration and therefore with neuronal death. In our group, we are interested in studying neuronal death and particularly apoptosis and the mechanisms regulating it in order to try to prevent it. So, among the different modulators of apoptosis, we are focused on the fast apoptotic inhibitory molecule, or FAME, that is a death receptor antagonist and therefore inhibits apoptosis. There are two different isoforms of FAME that are produced by alternative splicing. The short one that is expressed ubiquitously, but it's not able to prevent neurons from apoptotic cell death, and the FAME-long form that is in which we are interested in, that it's expressed exclusively in neurons and is able to protect them from apoptosis. And the way it does is, in one hand, it binds to the death receptors and prevents the activity of caspase 8, and on the other hand, it binds SHAP, that is the main endogenous inhibitor of apoptosis, and stabilizes it, preventing its ubiquitination and therefore its proteasomal degradation. By the stabilization of SHAP, it allows to inhibit caspase 3 and therefore apoptosis. Some time ago, we described that FAME-long levels are both in the mRNA levels or in the protein form are decreased with the severity of Alzheimer's disease in the hippocampus of these patients. And also we observed this same phenomenon in a mouse model of Alzheimer's disease, that is the APPPS1 model. So based on these results, we were interested in knowing the role of FAME-long in Alzheimer's disease. And the first that we did was to make a screening across different transgenic mouse models. And surprisingly, what we observed is that FAME-long decrease was only observed in mouse models showing tau pathology, with or without A-beta pathology. But we didn't find any alteration in FAME-long levels in models that only showed A-beta pathology, which suggested thus that the decrease in FAME-long should be associated with tau alterations. We decided to focus in the P301S mouse model, and we established a colony of this mouse model in the laboratory to study the progression of the tau pathology with the progression of FAME-long decrease. And as you can see, the progression of FAME-long decrease goes in parallel with the progression of tau pathology, in this case evidenced by the increase in phosphorylation. And it's evident at 6 months and goes on until 9 months of age. Importantly, these alterations appear before other alterations that can be significant for neurodegeneration, such as increase in GFAP as a marker of neuroinflammation, or decrease in PSD95 as a marker of synaptic dysfunction. So these are only evident at 9 months of age, while still not significant at 6 months of age, where tau and FAME are already altered, suggesting that it may have a potential role in the neurodegeneration progression. Then, and knowing in which molecular processes FAME is altered or participates, that if you remember, I told you that it participates in the ubiquitination, preventing the ubiquitination of SHAP, we observed in these animals that it's a trend when fame starts to decrease, but it becomes evident at 9 months of age, that we observe in these mice a global increase in protein ubiquitination, and also a decrease in Shia protein levels when the fame long decrease is already established. So based on this, in parallel to these in vivo studies, and with the aim of finding novel molecular interactors of fame long that could shed light into its function, we performed in vitro studies with neuronal-like PC12 cells that express fame, and we extracted protein, immunoprecipitated, and went through a mass spectrometry study from which, among different proteins and molecular, and biological processes, Alzheimer's disease and tau came up. We then confirmed this potential interaction by performing in vitro immunoprecipitation between tau and fame, and confirmed that fame and tau immunoprecipitate, and interestingly, the isoform long of fame, so fame long, interacts better with tau than the short form. So based on these results, we wanted to find a significant relevant biological function from this interaction, and knowing again the molecular processes in which fame participates, we analyzed tau ubiquitination in the presence of fame. And we observed that in the presence of fame long, there is almost an abolition of the ubiquitination of tau that is not present in the, that is not observed in the presence of fame short, indicating that this is a function exclusively of fame long. Also, with the aim of knowing which are the triggering factors that induced the decrease in fame long, we analyzed in vitro also the effect of phosphorylation of tau in fame long levels. And as you can see here, we overexpressed a wild-type form of tau or a mutated form of tau that is more prone to phosphorylate, and together with GSK3 beta expression that potentiate even more the phosphorylation of tau. And we observed that the lower levels of fame long are observed when GSK3 beta is present, so meaning that it depends on the phosphorylation of tau or in the active itself of GSK3 beta. So these results prompted us to postulate this model in which fame long under normal conditions would prevent tau ubiquitination and therefore tau aggregation. But in Alzheimer's disease conditions, when tau phosphorylation is increased, it would induce a decrease in fame long, and then it would lose the protection on tau ubiquitination, and therefore allow more tau aggregation that would contribute to the Alzheimer's disease pathology. I have to say that we have no idea of which biological meaning has this inhibition of tau ubiquitination, if it's just a way of preventing its degradation or if it has other signaling functions. So in order finally to confirm whether fame long is able or not to modulate tau pathology in vivo, we are now performing an in vivo experiment in which we are injecting adeno-associated viral vectors overexpressing fame long in the hippocampus of 6 months old P301S animals. This is, if you remember, is the time when fame long starts to decrease. And we are analyzing at 9 months of age the behavioral, biochemical, and functional parameters. And results are about to come on the behavioral analysis in the next weeks. So I'm sorry, I cannot tell you any more about this. But I can conclude that fame long levels are exclusively reduced in mouse models showing tau pathology, that this decrease occurs before the onset of the neuronal pathology in these mice and correlates with alterations in ubiquitin and apoptosis pathways, that tau and fame long interact in vitro, that fame long reduces tau ubiquitination, that the phosphorylation of tau reduces fame long levels in vitro, and that the ongoing in vivo experiments hopefully will allow us to determine whether fame long is involved in the progression of tauopathies and if it can be postulated as a novel therapeutic target. And I would like to thank all my group, especially Raquel and Alvaro, who are performing this project, and obviously all our collaborators, our funding agencies, and all of you for listening to me. Thank you.
No? Wait for the poster then. Thank you very much, Monse. So we go to the final talk today is by Inmaculada Cuchillo Ibáñez, and she's a postdoc at Javier Saez Valero Group at the Instituto de Neurociencias, Universidad Miguel Hernández, CSIC, and CIBERNED. Her poster is number 60. Thank you very much, Inma.
Well, thank you very much. Everything I'm going to present here is made by Matthew Leno, who is our predoctoral student in the lab, and it is about the ApoE protein and the cerebrospinal fluid in Alzheimer's. Well, ApoE is the main cholesterol carrier at the brain and the CNS, and is mainly produced by astrocytes, but also by microglia, and in some conditions of stress by neurons. ApoE is a glycoprotein, and glycosylation is a post-translational modification that binds different sugars at specific sites of the protein. And glycosylation affects ApoE function and ApoE capacity of binding the receptor, and also its hydrophobicity. In humans, there are 3 isoforms of ApoE, ApoE2, ApoE3, and ApoE4. And they all have differences in the properties of lipid binding, but also in the affinities for the receptors. ApoE2 is considered as protective, and one single copy of ApoE2 protects against Alzheimer's by 40%. ApoE3 is neutral, and ApoE4 is the Alzheimer's risk factor. Yes, carrying one single copy of ApoE4 increases Alzheimer's risk by 3-fold, and carrying 2 copies of ApoE4 increases by 12-fold. The differences between ApoEs are in these sites, in these amino acids. They can have 2 cysteines, or a cysteine and an arginine, or both can be an arginine. And ApoE4 has 2 arginines. And because of this, ApoE4 is unable to bind to form dimers, because it hasn't got cysteine, so it cannot form disulfide bonds, which are the link for these dimers. And to form dimers is important for the activity of ApoE. Well, the aim of this study is to study the protein ApoE4 itself at the cerebrospinal fluid. You all know that in this fluid is where we can find the biomarkers for Alzheimer's, as the A-beta, tau, and neurofilament. And we are interested in the ApoE monomers and ApoE dimers. Well, we have employed 2 cohorts. One is from Sweden, from Gothenburg, and the other one is from Barcelona. They are similar in the characteristics. The one from Sweden is a bit larger, is 59 samples of females and males, and the one from Barcelona is 39 females and males. We have the genotype 3, and 3, 4 for control samples, and for Alzheimer's we have 3, 3, 4, and 4, 4 for Alzheimer's. We don't have any 4, 4 as control because it's quite difficult, and all of them has biomarkers for Alzheimer's. Everything I'm going to show here is made by Western blots. Well, when you see ApoE in a gel, ApoE monomers, we always see 2 bands. The upper band is considered the mature form of ApoE because it's fully glycosylated. And the lower band is considered less glycosylated, but it's also called as immature form of ApoE, or the native form of ApoE. When we quantify this in the Alzheimer's control samples, we always find that these 2 monomers of ApoE are more abundant in Alzheimer's disease. When we do the ratio between the mature and the native form of ApoE and different genotypes, we see that this ratio is lower in Alzheimer's and is higher in controls. So this could suggest that in the pool of ApoE at the CSF of control samples, the mature form of ApoE could be predominant respect to the native population of ApoE. On the contrary, in the Alzheimer's samples, both populations contribute more or less in the same weight. So you have native form contributing to the population at the CSF in Alzheimer's. When we evaluate this ratio in the Alzheimer's population, also including the 4 and 4 genotype, we see that ApoE4 has also a high ratio of monomers of mature respect to the native, which reminds a bit two controls. So ApoE4 is behaving as a control ApoE, where the mature form of ApoE4 is contributing more than the native population of ApoE4 in Alzheimer. We did a very preliminary study in frontal and temporal cortex at different plaque stages, so different stages of neurodegeneration, and we observed that the native form was present in control and in all plaque stages. But the mature form of ApoE was only present in control of early plaque stages, but it was very weak in late plaque stages. So these results are pointing in the same direction of the CSF samples, where in Alzheimer, the predominant ApoE could be the native or immature form of ApoE. About the dimers, we found dimers in control and in Alzheimer samples. The question was, is ApoE contributed to these dimers? And the answer is yes. When we use a specific ApoE4, we found that, the answer is yes, depends on the sample. In control samples, ApoE4 doesn't participate of these dimers, but in Alzheimer, it is participating in these dimers. When we do a native blot and we analyze the ratio of dimers between monomers, we see that for the genotype 3 and 3, there are abundant dimers in control and in Alzheimer samples. When we analyze the genotype 3 and 4, we see that in Alzheimer, there are more dimers than in control samples. And also, the genotype 4 and 4 has these dimers, which is a bit unexpected because it hasn't got these cysteines. So we know that these dimers are linked by disulfide bonds. So if we reduce this link by using beta-mecaptoethanol, which is reducing these links, we found that in control samples, in control samples, we don't have any dimers because they depend on this kind of bonds. But it is, but it is, but it is present still in the Alzheimer samples. And ApoE4, it is also participating in this kind of aberrant dimers that do not depend on disulfide bonds. So when we quantify this, these different dimers are only present in Alzheimer but not in control. And when we stratify this by the genotypes, we see the same. These resistant dimers are only in Alzheimer but almost nothing in control. And in Alzheimer samples, they are in all the genotypes, including the genotype 4 and 4. Well, so in conclusion, we see that there is the levels of the monomers is higher in Alzheimer respect to controls, and that there is an imbalance between these glycoforms, the fully glycosylated, which is the mature form of ApoE, and the immature or native form of ApoE. And about the dimers, there is only this kind of resistant dimers in Alzheimer, and ApoE4 is also taking part of these dimers, even if it hasn't got this cysteine. So it's another kind of link that we want to study. No? So ApoE is forming this kind of aberrant dimers, specifically in Alzheimer. Well, I think I'm going to be a bit fast. I want to just thank you to the lab and also to the lab in Sweden who just gave us these samples, and the lab from Barcelona that gave us another other cohort, no? And also from the lab from Nahuatl for another kind of samples from rat, and I haven't shown these results, and that's all. Thank you very much.
Thank you very much, Marta. So, is there any question? Yes, Pepe was.
Yeah, very interesting. I wanted to ask, which is the chemistry of the chemistry forming the bond, the non-disulfide bond?
Yeah, we don't know yet. This is something that we have to study because Well, it could be that because the population of mature and immature form are present in the CSF, could be that there is a dimer formed by the native, which is not glycosylated, and the mature form. And that doesn't depend on the cysteine. It could be another kind of covalent dimer.
Okay.
Because it is resistant to, I mean, we don't think it is a hydrophobicity link because it will disappear in the Western blots. No?
Okay.
So probably another covalent link.
Okay. Thank you.
Question? I saw some hands over there, no? So, thank you very much. I think we've done very well. Yes. So 5 minutes in advance, this is incredible. So, I would like to finish thanking all of you because you did really a good job, and your research and your presentations are really perfect and an example to follow by other young researchers that we will really like you to we encourage you basically to ask for presentations in next meetings so that you can as well train your training capacities, abilities, and, you know, to show off a bit. So thank you very much, and please send more, you know, participating as speakers in next meetings. Thank you very much.